The wild wheat population of Ammiad is located in northern Israel, close to the Sea of Galilee, at the center of distribution of the species. The area was declared as a natural reserve for in situ conservation study and was sampled since 1984 from 100 sampling points that were marked with pegs and GPS coordinates across 4 linear transects and different ecological niches. This project was designed to allow us to ask questions on how wild populations change over time and space in their natural habitat under occurring climatic changes. We have genotyped a collection of more than 800 samples over 36 years using DNA sequencing and saw that the population is diverse and divided to clusters of genotypes, represented here by different colors which appear to correspond to the ecological microhabitats. This pattern was repeating year after year with a remarkable stability over time. For example, there is a specific genotype that resides under this tree and haven't changed throughout the study. But what causes this spatial and temporal stability? Is it just because the wild wheat does not disperse much? Or is there some selection or local adaptation involved? We ran simulations using different parameters of seed dispersal, outcrossing rate, seed bank, and plant density. These results show that under the most realistic parameters, the observed stability does not overlap with the simulations. So, unless dispersal rate is extremely low, the spatial and temporal clustering of the population could not be explained by neutral processes alone. We learned from this study that in situ conservation of wild plants biodiversity at sites like Ammiad can be achieved in a relatively small area to preserve high diversity and valuable genetic resources for crop plants.